All right, chapter 1. Before we get started, I'm going to recommend that you have a copy of the PowerPoint next to you or in a window that you can take notes directly on the slides. Since it's a recorded lecture, you can pause me, you can rewind me, you can fast forward me if you like. And so I'm going to go through these reasonably quickly because you have those options and abilities. So chapter 1 is an overview of really the whole course, not just Biology 1406, but really also Biology 1407, and the whole concept of what science is, what biology is, and then a couple of things that you definitely need to memorize and focus on in this chapter. All right, so biology. Your goal is to understand. And part of understanding in biology is that you have to know some facts. You have to memorize some facts, and you have to be able to recall those facts. I think that's what makes biology hard for students is the number of teeny tiny little facts and precise definitions that you really need to know in order to really be able to build a base of knowledge in biology. Because ultimately you're going to have to pull those details and those facts out and be able to put them together to make larger concepts and to be able to figure out new problems and new situations by relying on those facts and those rules and those fundamentals that you'll learn in this course. So I have to say that memorization is part of it. And that's not a dirty word. You have to memorize some things and just make it to where they come back to you very quickly if you're going to be able to move forward and be successful in biology. All right, themes. Themes that organize biology. One is evolution. Evolution is a huge theme. We're not going to talk a ton about evolution in this course other than just in some general terms. You'll learn a lot about evolution in Biology 1407. And so because that's the part of the course that studies biology at the population level, we're going to be studying biology mainly at the cellular level. And it's not that evolution doesn't apply at the cellular level, but we just don't start with that topic. We start mainly with cells and cell theory. We start with cell structure, not just DNA structure, but cell structure and function, and the concept that structure determines function. You really need to have Biology 1406 and 1407 to get the full picture. So if you just take 1406, you're going to be missing a big piece of it. All right, the process of science. Science in general includes what we're part of the natural sciences. Those are the fields of science that relate to the physical world. So physics, chemistry, biology. In some ways, there's just an artificial separation of these fields. In other words, biology involves chemistry and physics, just like chemistry involves biology and physics involves biology and chemistry. I mean, there's no... on Earth, all these things interact. I guess if you had a world that didn't have any life, then biology might not be important. But on Earth, and that's the only world that we're going to be talking about, all three of them interact. And all three of them are part of one and the same. So one of the rules, though, is that in science, you're restricted to explanations that can be demonstrated and can be repeated and rely on certain rules of logic. And there are things that just don't fall within that realm. It doesn't mean they're not real. It doesn't mean they don't exist. It's just that science cannot address things like God, I guess what you would call magic, or things that are what we call supernatural. we don't address really, we have a hard time addressing whether there are ghosts. I'm sure you enjoy watching shows, you know, ghost hunters or things like that, ghost adventures. I enjoy them too, but it's more of an it's not really science. I know they have a lot of equipment that seems very sciencey, but at the end of the day, they never can say for sure if they have evidence or not, and it's just beyond the realm of science. And so we stay away from those things, not because they may not exist, but because that science is limited and we don't address those kinds of things. All right, in science, there's two types of reasoning. One's called inductive. Inductive reasoning is really how scientists do their research. It's where you try to gather data and try to come up with a conclusion. You look at your results, which are little pieces of data, and you try to come to a general conclusion about how the world works, how life works, how chemistry or physics works. Inductive reasoning is how real scientists do science. Deductive reasoning is more about how you learn about science. As a student, mostly what you do is you learn the rules, the general principles, and the laws, and you apply those to figure out solution or predictions or specific results. So deductive reasoning is usually, as a student, that's what you're learning. You're learning all the principles and laws and rules that are already known or believed to be true, and to form that foundation of science. Inductive reasoning is where you're trying to come up with new discoveries. So deductive reasoning is probably pretty comfortable for you because you have a textbook full of all the principles and laws that scientists have figured out already, and you just kind of learn those. You don't really learn the research that came to lead to those rules. But inductive reasoning is actually what scientists do, and that students have trouble with inductive reasoning sometimes. That's more of what we're going to be doing in lab. In other words, I'm not going to tell you what the conclusion is because you're going to be looking at little pieces of data and seeing if you can make a logical argument to a conclusion. So deductive reasoning is what you learn from your textbook mostly. Inductive reasoning is really what we're doing in lab to gather data and try to come up with a conclusion. The scientific method. It's typically shown in this way, and if you have quiz questions about this, you can use this format. I never once sat at a desk and followed these steps. I did research science for many years, but what this image is a way for scientists to try to explain to maybe a non-scientist how our brains naturally work. We look around, we make an observation, we say, that's interesting, that's weird. And then we ask a question, I wonder what caused that? And then we say, well, maybe it is, and that's the hypothesis, a hypothesis that tries to answer the question. And then we test it out. If that's correct, then I should be able to do this. Then we do the experiment, we see if we got what we thought or not, and then sometimes that makes us revise our hypothesis and do it again. I would say in general, it's not this formal. I know in the science fair, if you have ever participated in the science fair, they make it very rigid. And once we are ready to publish our results, we do kind of organize everything and make it more organized. But when you're in the process of science, it's much more just like you would solve any problem, is think it through, test something, see the result. Maybe you have 4 or 5 tests going on at the same time. So this picture is very formal, but it's not that rigid. But in any case, there has to be a logical process of your thinking. That certainly is true. But this is what we call the scientific method. A lot of science happens just by luck. So you can't eliminate what we call serendipity. Serendipity is where you are doing one set of experiments and you get a result that doesn't match anything that anybody's ever seen before, and you end up going off in a whole new direction with a whole new question and actually discover something that you didn't even know existed before. So that's happened to me several times, where we discovered something that we didn't even know we were looking for, but when we saw results that were just so unexpected, we had to start questioning some of our assumptions. And it led us to new discoveries, and we didn't mean to discover that. It was what we call serendipity. So a little bit of luck. And knowing when the results are telling you something new. Also trying to figure out when the results are just because you screwed up. So that happens a lot in science too. I mean, the reason I know what I know about how to do research science is I've screwed up everything at least once. So it's a slow process of learning how to do research and learning how to be an expert in one particular area, because you've screwed those experiments up so many times that now you're an expert. That's what being an expert probably is in any field, is you've already made all the mistakes, and you know all the what's going to happen if you do this or that. But the scientific method is, I mean, this is a very formal presentation of it, but it's just the way that scientists think about a problem. A good hypothesis is where you try to figure out what's causing something. It has to be testable. That's where the supernatural questions stop being science, because there's no real test for if there's a ghost. Maybe there is, maybe there isn't, but what's the test? And it has to be falsifiable. In other words, you have to do a test. I think in grade school, it's usually called, it has to be a fair test. There has to be a result that could prove that your hypothesis is wrong, and there has to be a result that supports your hypothesis. So it has to be a fair test. And like I said, when you're doing research, it's an inductive process. You're building upon little pieces of data, and so it's very inductive. You don't know the result. You don't know the conclusion while you're in the middle of the experiment. So when we do research, like when we try to do research in our lab class, now, yes, I already know what the result is going to be, but we're going to pretend like we don't, like this is real research and we don't know what the result's supposed to be. So you have to repeat the experiment over and over again to make sure you're not screwing it up. And then when you keep getting the same result over and over again, then you can say, okay, this is really the result, so what does it mean? And so that's the thing. We don't know what the conclusion is ahead of time in real science. So when you ask me in lab, is this the right result? I want you to think about it more from the perspective of, how would you know if this is reliable? Have you done all the steps correctly? Have you checked everything? Have you repeated it over and over and got fundamentally the same kinds of results? So those are the things that we do, because we don't know what the conclusion is ahead of time. Otherwise, we wouldn't be doing the research. We wouldn't be finding new things if somebody else already knew the answer. Variables. We have two variables, and this is just like a graph. When you do a graph in math, on the x-axis, that's what they call the independent variable. A lot of times in science, that could be time, could be the independent variable, and then, but it could be something else. And then on the y-axis, when you're graphing it, the dependent variable is the effect. So if you're tracking the progression of a disease in a population, for example, time is on the x-axis, and then the y-axis can be number of cases, and you can see it going up and down. So, but cause and effect. So that's a good way to think about it. The x-axis is the independent variable. The y-axis is the dependent variable. Right now, we're just leaving it very general. But when we get into lab, you'll see we'll use some actual examples and we'll see how it fits. Controlled variables. Controlled variables are not the control group. So you have two groups in your experiment. You have your experimental group and your control group. So your experimental group might be a group that's getting a vaccine, and the control group that did not get the vaccine. And then you're tracking both groups and seeing which group gets the disease more often over time. The control variables are something you apply to both groups. The control group and the experimental group have certain variables that you're trying to control. So maybe you want to make sure that the two groups don't have any differences in age or don't have any differences in health conditions. You want to make sure that those things are the same between the two groups. So that the only thing that you're varying between the two groups would be the experimental variable, which might be giving one of the groups a vaccine and not giving the other group a vaccine. But any other variables need to be consistent across both, like age, health conditions, and those two things. Those are the controlled variables, which are constant across both groups, the experimental group and the control group. That sometimes is confusing to students. All right, let's talk about facts. Facts, we can never prove anything absolutely. So we say facts, but we know in science, we know that nothing is really a fact. I mean, it's as close as we can ever say that something is absolutely true, but we know nothing is 100%. You can never absolutely prove something, which is annoying at best. But you can continually collect data that supports something. So I can continually collect data that supports that all life is made of cells. Cell is the basic unit of life. It'd be hard for me to imagine anything we could find that would disprove that statement, that a cell is the basic unit of life. But we can only continually support that. So that's the language we use. We repeatedly support something. We can't absolutely prove it. So that's where I say scientists accept something as equivalent to a fact, but nothing can ever be absolutely proven. So that's annoying. A theory is even bigger than that. A theory is a whole bunch of, I guess, facts or things that are supported by data. Would be better to say it that way. And it's supported by a very large body of evidence. So facts or things that are supported are like small things, individual things, and a theory is a much bigger concept. So in the end, it's not a huge difference between the two, although, what is conventional thinking can change. So we usually think things are facts, and then over time things do change in science. That's where you have to say nothing is absolutely proven. So there's always that room that something could be wrong, but we go with what we believe to be true, and then if we find evidence that disproves something, then we shift that. So that's true. That's a reality you have to live with. Some things that were taught to me as being this is the conventional wisdom, we think this is correct, have changed somewhat over the course of my career. So nothing like evolution. Like evolution is a theory. So that's supported by a large body of evidence. It's very hard to imagine how that might be disproven. But like little things within the theory, little facts, like how closely these two organisms are related, that can shift or change through new discoveries and such. So most everything is really theory, truly speaking, not fact. Okay, enough about that. Basic science. Basic science is what happens in university laboratories. Also, basic science happens in private companies. Basic science is the goal. The goal of basic science is just to learn, just to find answers to questions and learn as much about something as we can. A lot of times in basic science, it's hard to imagine what the application or importance is going to be, but we trust, and we have experience that this is true, that some of the knowledge that we get will later become useful to us. Not all of it, but some of it. So we keep churning and creating a base of knowledge, and some of that ends up becoming important for future discoveries and future applications. Applied science is really what picks and chooses from the basic science and uses that knowledge to solve problems, like creating a new drug or a new treatment for a disease would be a common version of applied science that we see in biology. So basic science, I mean, sometimes you're just researching the structure of an enzyme in a pufferfish. And it's hard to imagine, why would anyone care about that? But when all of those kinds of things are learned, it sometimes, like I told you with serendipity, sometimes that enzyme from pufferfish, later on we find out that that's the cure for XY disease. I mean, those things happen. Or sometimes we realize we can use these things for a certain purpose. So, for example, they found some enzymes that are released naturally by bacteria, but then they figured out that they can be used to break down plastic. So it becomes a solution, or the beginning of maybe a solution to the issue of, conservation, of dealing with the large amount of plastic waste that we have in the world. So by studying just all kinds of things, and it's amazing the things that people spend their whole career studying, this one enzyme, this one organism, this one thing, and then some of that is useful in the future. It becomes useful when people realize how it can be used. So that's kind of the fun part. The first lab I worked in, we worked on cyanobacteria, which is blue-green algae, but it's really a bacterium. And that my PI is the professor, stands for primary investigator or principal investigator, but he was the professor, and it was a research lab, and he went on to work with that organism creating biofuels. So, it progresses. It's a slow process for sure. So when you work in research science, and like I said, it's a very inductive process, not so much a process that you're familiar with. When you have a discovery, like I said, we discovered something new in that cyanobacteria just by luck, and maybe someday I can tell you about that. Then you publish something. You publish a paper, and papers are published in what we call peer-reviewed journals. And you need to write this down. The top journals in biology are Nature, Cell, if you're in molecular biology, so Cell, not a surprising name. And then in general, in all of science, there's a journal called Science. So those three are really your top, what I would call your top tier. It's kind of the equivalent of like Harvard and Yale for top tier colleges. You have top tier journals. And so you want to get published there, but we didn't publish there. And then there's like mid-range journals. We got published in what's called Proceedings of the National Academy of Science. That's a middle range. That's a nice, solid middle range journal. Actually, a little on the upper edge of journals, I would say. But anyway, so we got published there. I published a paper in a journal called Genomics. And then I do have a Nature paper, but I'm like, my name is listed like 200ths. There was hundreds of authors on that paper because it was part of the Human Genome Project. And so I'm way down on the list on that one. But anyway, so you get published in a peer-reviewed journal. And that's the standard for getting a PhD in science. You have to have publications. And so when you go to university, your professors are trying to get publications. They may or may not be good teachers, but their main where they earn their living is through their research and their publications. So so when you write a paper, you have a certain format. You start off with what's called the abstract, and we're going to practice this in the course. You're going to get to write one paper. And just kind of mimic how that would be. The abstract is like the summary. Most scientists just read the abstract. On most papers that I read, because I do have a subscription to Nature, if you're interested, I can send you some articles from that, just so you can kind of see how those look. I do have a subscription to Science, and I do have a subscription to Cell. And so anyway, when I get those, and I get some of those I get digitally, some of them I get in paper form, and I just skim through the abstracts because not all of them are interesting articles to me. I have a specialty in molecular biology and genetics, human molecular genetics, and so I kind of pick through and look for those kinds of things. But anyway, so the abstract is the summary of the whole thing. Most scientists just read the abstract because we're lazy, I guess. And then if there is something that's really catches our interest, we might read the whole thing. So the rest of the article is the introduction, the full length, the methods, results, discussion, and then a bibliography. And so we're going to kind of mimic this we're going to write up a little lab paper and kind of mimic this structure. So you get a little of that sense. All right, what are the properties of life? This is what your book put as the properties of life. I could make maybe a different list, but it would be fundamentally the same. Order, circle that one. Order is going to be a big deal for us in this course. We're going to figure out how the molecules make up the cells, how the cells make up the organs and tissues and organisms and such. So there's a hierarchy of order. And I'm going to instruct you to do a little bit of book reading on that topic. Response to stimuli, reproduction, growth and development, those are going to be more focuses of 1407 biology. So not that we won't touch on some of those, but metabolic processes and energy processing, those are going to be big ones for our course. And so some of these are going to be more important. I would circle, if I were you, order and energy processing becomes a huge part of this course. The first one and the last one. We'll touch on some of the ones in the middle, but I have to build a fundamental, a basis for you of vocabulary and basic understanding before we can really dive into some of those other topics, which will be in 1407 and then also in future biology courses. All right, so order is a really important topic for this course. Like I said, response to stimuli, not so much. Reproduction, growth, and development, a little bit. I mean, we'll be talking about how a cell and a little bit about sexual reproduction, how cells divide, a little bit of that. Not a lot about development. All right, energy transformation and energy processing is going to be huge for us. I know that doesn't sound like biology, but it actually is a huge part of biology, is the energy, that how energy is transferred and transformed in the organisms. We're going to be doing it, looking at it at the cell level. So here's where I want you to go to your textbook. When you're done, make yourself a note. And make sure you have a definition for each one of these. This is the levels of organization of life, of biology. And it starts at the atom. Of course, there are smaller components of atoms, but we're going to start with the atom. And we're going to learn in Chapter 2 a little bit of chemistry, a little bit about the atoms, a little bit about molecules. In Chapter 3, we learn about macromolecules, which is a beast, I'm not going to lie. And you need to have a definition for each of these terms, but this is the order. Atoms join together to make molecules. Molecules, very large molecules, are called macromolecules, and they construct what we call the organelles, which are the little parts within a cell. And so organelles are part of cells, and cells that are all of the same type and do the same kind of function make up tissues, and multiple tissues come together or work or combine to form organs, and multiple organs create organ systems in the body, and then the organ systems in the body make up the organism. So like the human is one organism, and an organism has, a human has a respiratory system, a circulatory system, a urinary system, a reproductive system. I'm forgetting some, but all those systems make up the organism. And then multiple organisms of the same species make up a population. And different populations of different species that live together in one area are called a community. Then when you add in what we call the abiotic parts of the environment, like water and rock and sun, that makes up the ecosystem. So you probably learned about some ecosystems in middle school. You have the tundra, or the grassland prairie, or the Arctic, or those kinds of things. And then all the ecosystems make up the biosphere. And the biosphere is where we stop. That's the world. That's Earth. We're not going to try to go beyond Earth. Whether or not we have any data of life beyond Earth, that's just not going to be the scope of this course. So you do need to memorize the names and the order in which these occur. And so the atom is the smallest and the biosphere is the largest. So make sure you memorize them in order and know the definitions of each of them. And in this course, we're really only going to, let me see if I can get a pen going here. We're going to look at atoms, molecules, macromolecules. We're only going to go up to cells in 1406. So you do need to know the whole list. But in terms of detail, we're going to spend the course going into detail on just these ones. All right. And then in 1407, you'll go into detail. Usually you start at populations. You do some ecology and then some anatomy and physiology. So you touch on some of these others. And so you do need to know for chapter 1, there's the whole list in order with definitions, but just as an overview, this is these 5 at the top is where we're going to start going in real detail in 1406, and I'm going to teach you the vocabulary and the fundamentals so that you can really have a good basis. This is what's in your book. So make sure you go through this and spend some time with that. All right, types of cells. I bet you know this already. There's 2 types of cells in a very rough separation. The 2 fundamental types of cells in the world are prokaryotic cells and eukaryotic cells. Prokaryotic cells would be things like bacteria, or another group that we call the archaea. Those are prokaryotic. They don't have a nucleus, and it says they lack a nucleus in other organelles. They actually have structures or organelles called ribosomes, but they don't have most of the organelles. They specifically don't have a nucleus. They do have DNA, but they don't have a nucleus. A nucleus means the DNA is surrounded by membrane. And then the eukaryotic cells is where the DNA is surrounded by a membrane. That's called a nucleus. So this picture shows bacteria, all right, invading human cells. The human cells are shown in the yellow. Of course, the human cells are much bigger than the bacteria, so you don't get to see the whole human cell at once. But you can see the individual bacteria. There's 2 over here on the left, top left, for example. All right, this is how we think. Oh, that's weird. Let's see if I can, let's see if we can look at this. This picture is, oh, it's cut off. Well, you have one in your book. This is called the Tree of Life, or the evolutionary tree of life. And the branches should meet together at the bottom here. There's like a trunk on the tree. So sorry about that. I didn't notice that. But anyway, this just shows how the different groups of organisms are related. So bacteria and archaea, those are prokaryotes. So all of this, and really the original tree starts with, we think, prokaryotic organisms. Here, I'll finish it for us. Here, these come together. And then we think, this isn't actually completely accurate, but we think that a combination of archaeans and bacteria came together to form the first eukaryotes. So I would probably draw this a little differently. But in any case, the eukaryotes, we are, they put a little star where the animals are. Of course, humans are a very, very small proportion of animals, but we're over here. So the eukarya is the what we call the domain. It's called domain eukarya. And that is the grouping of organisms that has all the eukaryotic organisms. So you can see all the animals, all the fungi, plants, and then there's some other things. And then these guys that I circled in red, these are prokaryotic, but we don't have a domain called prokarya. We just have a domain called bacteria and a domain called archaea. So you do need to memorize the three domains, the names of them. So domain bacteria, domain archaea, and domain eukarya. And then within the eukarya, I think it's helpful that you remember that the animals and the fungus and the plants are in there, and then some other stuff. But those are kind of the three big groups. So when you take a microbiology class, you'll focus quite a bit on the domain bacteria and domain archaea. And then typically you'll take a cell biology class that focuses more on the eukarya. But the main division is the prokaryotes from the eukaryotes. All right. And I want to end with a little discussion. This is more of a practical discussion of what we call models. Models in science. Models are some types of representation of things that happen in, for us, it'll be biology. It can be diagrams, and that's what you'll see in your textbook. It can be three-dimensional objects, models that you actually see like in lab, like a plastic model of the human body or something like that. We also use computer programs and math equations to model events, random events or non-random events. So we won't use those so much in this course, but in future courses you probably will. I've used computer models to mimic how different things affect how a neuron reacts to something, or how evolution affects a certain population. So computer programs, which usually have as part of the program mathematical equations, can be models. But anyway, for you it's going to be a lot of diagrams in the textbook, and then when you do get back on campus, three-dimensional objects in lab. The thing about models is they typically are kind of simplified. In other words, they focus on one thing and ignore other thing. They don't tend to, they're not real to life, typically. And that can be confusing because you may not know which part is being emphasized and which part isn't being emphasized. So I'll give you an example. This is a model of the human heart. This actually came from, this is a diagram from a textbook. And so you can see it's showing how blood flows from the body to the right atrium, to the right ventricle, to the lungs, and then back from the lungs through the left atrium, through the left ventricle, and back to the body, and then it loops back. So it's just teaching the order through which the chambers of the heart through which the blood flows in order. But it doesn't, obviously it's not meant to indicate that the heart is a square, and it's not meant to indicate that the left side is red and the right side is blue, or, so there are things in it that aren't meant to be true to life. You probably know that, though, because you kind of know what a human heart looks like, so you automatically kind of know what is implied here as what is being taught here is simply the flow of blood through the chambers. But if you never have ever seen a heart before and you're looking at this, you might make some assumptions. You might think, okay, the heart is a square. Okay, and I'm being, of course it's ridiculous, but I'm saying when you're looking at something you've never seen before, which in this course you will, sometimes you do have to ask those questions. Okay, what am I supposed to be learning from this and what not? And if you can't tell, that's something you ask me, all right? Another problem that students have is with scale. Because when you're looking at models, especially book pictures in the book, you can't tell how big something really is. So I give you this example. There's 4 images of what I call men, I guess. There's the man, I don't know, the bathroom sign, I guess. These men are from a board game. And then this is a bobblehead man. And then down here is the best quarterback ever, Troy Aikman, of course. And so the picture of Troy Aikman. It used to be the Cowboys quarterback when the Cowboys were good. Okay. Anyway, so you have models of men, but in your book, they'll be pictured at different sizes. Now, the thing is, that I think the men that are like from the board game, they're only like this big in real life. I mean, they're like an inch. So that's actually the smallest. But in this picture, they're the biggest, or at least as big as some of the other things. The picture, the bobblehead and the sign for the bathroom are probably about the same size, maybe 8 inches or something like that. And of course, Troy Aikman is 6 feet tall. So what I'm saying is that when you have experience with things, your brain automatically puts them in the right scale. Like, I know that a little board game piece is this big. I know that a bobblehead is about this big. I know that Troy Aikman is about 6 feet tall. The problem is when you have something you're not familiar with. So I'll give you an example. So this is kind of like what you deal with when you're looking at a textbook. So now which one is bigger? And your brain wants to put things in scale. It really does. It helps you to learn it. So when you have a problem like this, again, this is where you ask me. Okay, I'm having trouble. You might come to me and say, I'm having trouble with this image. Is this bigger or smaller? How does my brain organize this? Because at the top left of your screen, you see I outlined it in blue. It's a potato cell. And it has some structures on the inside, but the light blue outline is one potato cell. On the top sort of right or center, you have a parasite called a fluke. On the bottom left, you have 2 cheek cells. And on the bottom right, you probably recognize the coronavirus. But when you look at the way I've presented it here, it's almost like the coronavirus is the same size as one of the cheek cells, or the coronavirus is the same size as the entire potato cell. But in fact, it's not. A virus is much smaller than any of these items. But when you see them like this, you don't have a sense of scale. You don't have a sense of actual size. So if you find that I just want you to be aware that you might be struggling with that and not even realize it. So when you're looking at some of the images and such, hopefully you'll be able to put things in scale. And that's where that hierarchy comes into play. An atom is smaller than a molecule is smaller than a macromolecule is smaller than an organelle, which is smaller than a cell. Of course, that doesn't help you with a virus. A virus is teeny tiny. But anyway, so virus would be bigger than a molecule, smaller than an organelle, or maybe about the size of an organelle. That's where virus would kind of go in terms of the size. So anyway, okay, well, I hope this helped, and that's the end of our Chapter 1 lecture.